Hi there, welcome to AP BioFun with Dr. D. Last time we talked about the major phases of the cell cycle, interphase and mitosis, and what is happening in each of these phases. Today we're going to talk a little bit more about how the cell cycle is regulated. And to do that, I'm going to introduce two concepts, two players really. They're called SPF and MPF. SPF stands for synthesis promoting factor, and MPF stands for maturation promoting factor. And pretty much nobody calls them that anymore, but it is worth exploring how they were discovered because it is an interesting exercise in experimental design and how we interpret experiments. So, in the first experiment here shown in this model, researchers took a cell which was in S phase, so it was in the middle of duplicating its DNA, and a cell that was in G1, and they forced the two cells to fuse with each other. And when the cells fuse, their cytoplasm become one, but their nuclei stay separated. And what they found is that the G1 nucleus immediately entered S phase and started synthesizing its DNA. Similarly, in a second experiment, they took an M phase cell and fused it with a G1 cell. And in this case, the G1 nucleus began mitosis. It started condensing its chromosomes and aligning them on the metaphase plate without having duplicated them. So that cell had not undergone DNA replication yet immediately entered mitosis. And the conclusion from these experiments that the researchers reached is that there was something in the cytoplasm of this mitotic cell that forced the G1 cell, that made the G1 cell enter mitosis, even though it wasn't ready because it hadn't duplicated its DNA. Similarly, there was something in the cytoplasm of the S phase cell that made that G1 cell start DNA replication immediately. And from these very, very early experiments, researchers concluded that there's some kind of factor that regulates entry into S phase and some kind of factor that regulates entry into mitosis. And they called them SPF and MPF. S phase promoting or synthesis promoting factor and M phase promoting factor. Well, years later, researchers realized what these factors are, and these factors are series of, guess what? Our old friends, kinases. Remember, kinases are enzymes that can put phosphate groups, they phosphorylate. Well, these kinases are a little bit weird. They're called cyclin dependent kinases because they're actually made up of two units. One is the actual kinase, the CDK, and another one is a protein called cyclin, which regulates the activity. So, cyclins are really what makes the second protein, the CDK, the kinase, active. Without the cyclin, the CDK is not active. And the two must be together in a complex to be able to be active, to function. So cyclins were proteins that were first discovered actually in sea urchins, and they're called cyclins because they were constantly appearing and disappearing, highly periodic appearance, and that appearance coincided with stages of the cell cycle. So these fluctuations, these ups and downs in protein levels were linked to cell cycle stage, and they were called cyclins. And on the right here, you will actually see an experiment showing you how a cyclin, in this case, it's actually the mRNA for the cyclin, appears and then disappears, and then appears again and disappears. Very, very periodic. Now, the periodicity of cyclins is achieved by controlling gene expression, so controlling the expression of the genes coding for the cyclins. And genes are either turned on or off, so transcription is controlled. The stability of the messenger RNA is also controlled in a cell cycle dependent manner, and the translation of the protein. Finally, the proteins are also being degraded. When we need them to disappear, they're being degraded. So it's several different points in gene expression. Pretty much at every point, you can control gene expression gene expression is controlled to make sure that now I'm here, now I'm gone. So the cyclins are highly, highly periodic. So again, the cyclins are the regulatory subunit. So there's two proteins, the cyclin and the CDK. And the cyclin is not the enzyme. The enzyme is actually the CDK, but the CDK must be bound to the cyclin to be active. So the kinase, the CDK, doesn't change throughout the cell cycle. The cyclin is the one that appears and disappears periodically. So we say there is a cycling of CDK activity. So CDK activity appears and disappears very periodically. And these appearances are driven by the appearance and disappearance of the cyclin proteins, as I told you. So number one, you see that the cyclin protein abundance. Now I'm here, now I'm gone. But they can be controlled in other ways. CDKs can be phosphorylated. The cyclin CDK complex, the two proteins can either bind or not bind. So one can control whether they bind. And there are also inhibitors that can inhibit the enzymes. So there are multiple ways to control CDK activity, but the important thing to remember is the CDK activity is periodic. So it is low, then it's high, then it's low again. So depending on which phase of the cell cycle it is. And the majority of these changes in the CDK activity are really driven by the fact that the cyclin proteins are either here or they're not here. And when they're not here, the CDK activity cannot, the CDK cannot function. There is no CDK activity. Now, there's also different CDK activities, and different CDK activities will drive different cell cycle phases. There's CDKs that are responsible for regulating the G1 phase and the G1 checkpoint. There's CDK activities that are responsible for regulating S phase. There's CDK activities findings that are regulating mitosis. So they're the major classes are the G1 CDKs, the S phase CDKs, and the mitotic CDKs. And guess what? The S phase CDKs are the SPF, the synthesis promoting factor, and the mitotic CDK, cyclin CDK activities, are MPF, the maturation promoting factor. So all these factors that nobody knew what they were years and years ago when they were isolated initially, now we know what they are. They're cyclin CDK complexes. Now, CDK regulated events are, and this is very important to understand, one way. They're only one way. And remember in last lecture, I showed you how that kind of happened. So event B cannot happen before event A has happened. And once event A is complete and event B starts, event B will make sure event A does not repeat. So event A ensures that event B happens. And then once event B starts, it inhibits event A from happening again. So things are going one way, A, B, C, D. There's no A, B, A, C, A, B. It's only A, B, C, D. So it's going one way. The mitotic CDKs regulate that one-way movement. And an example of this is that, for instance, the mitotic CDKs, the MPF, really disappears at the end of mitosis. And it doesn't appear until the next G2 phase. That means you can't do mitosis until you have done G1 and S phase. So you need to duplicate your DNA first. And here is a model that shows you this. So in blue, you will see the abundance of the mitotic cyclin. In black is the actual activity of the mitotic cyclin-dependent kinase, or the MPF, right? So you see how that activity peaks in early mitosis in M phase, and as cells progress towards G1, they're exiting mitosis, the activity disappears. And it is low, it is non-existent until next time, until we are right here at the G2M transition. So there's no MPF activity in G1, no MPF activity in S phase, no MPF activity in early G2. So cells need to do all of these things that they normally do in G1S and G2 before they can enter mitosis, right? We don't want mitosis to be happening before the cells are ready. We don't want G1 and then mitosis. We need to go G1, S, G2, and then mitosis. And this is ensured by the fact that the MPF activity is only available in late G2, early M phase, and as cells exit mitosis, it will disappear, and it will disappear again until the next cell cycle. Remember the direction, one way. And here is an illustration of how that actually happens. So in order to make the MPF go away, in order to make the mitotic cyclin CDK activity disappear, what cells will do is they degrade. They will chop up the mitotic cyclin protein. And remember, without the mitotic cyclin protein, the mitotic cyclin CDK activity is inactive. You can't just have activity just with the CDK. You need cyclin protein. So by chopping up, by degrading the cyclin protein, the mitotic cyclin protein, you are really destroying MPF. You're destroying the mitotic CDK activity. So when does that happen? Well, the moment the sister chromatids split apart in anaphase, the mitotic cyclins are degraded, but not before. So once the sister chromatids are split in anaphase, they're separated, that signals the end of mitosis. That signals that the cell is now almost done with division. So now it's time to get rid of the mitotic cyclins. And the mitotic cyclins are degraded after the chromatids are split, and they won't be turned on until the next cell cycle. So this way, the cell makes sure that once the chromosomes split, we're not going to try to split them again and again and again until we're ready. We don't enter the next mitosis until we're ready to enter it. Now, what is G0? Remember in the last lecture, I told you that at the G1 checkpoint, at the restriction point, the cell makes a decision whether it's going to enter the cell cycle or whether it's going to undergo apoptosis, whether it's going to undergo differentiation, or whether it's just going to sit there and retire. Cells that are not entering the cell cycle at the G1 checkpoint enter something we call G0. It's a phase in which the cells are withdrawn from active division. Okay, often cell in G0 can re-enter the cell cycle if it receives the right signal that it needs to grow and divide. It'll go from G0 back to G1. But if it goes from G1 to G0, in G0 that cell is not dividing. It's not going to be undergoing S phase. It's not going to be undergoing G2 or mitosis. So a little bit more about checkpoints. So what are checkpoints? Checkpoints really are points where the cell will check that an event is completed before it proceeds to the next event. Okay, we want to make sure DNA replication is complete before we enter mitosis, right? It also sometimes checks whether there's any kind of damage and defects. Is everything okay? Everything that's supposed to happen, is it happening the way it's supposed to happen? A checkpoint is triggered only when something is wrong. So let me just explain this again. Checkpoints are always active. That means the cell is always checking whether an event is complete and whether everything is done correctly. But when I say checkpoint is triggered, that means if something's wrong, the cell will stop. That's what a checkpoint means. So the checkpoint is triggered only when something's wrong. And there's checkpoints at several different places in the cell cycle. So what happens when a checkpoint is triggered? It's basically like you're riding the train, you pull the emergency brake, and the train will stop. Cell cycle arrest. So cell cycle arrest means the cell will stop. It will not proceed throughout the cell cycle. And that cell cycle arrest is temporary, okay? Because the cell will stop and it will attempt to correct the problem, okay? That's one thing that could happen to the cell. Now, Another possibility is that if the checkpoint is triggered, the cell undergoes apoptosis, which is programmed cell death. Cell will undergo apoptosis if the damage, the problem is really serious. If it's a huge problem, if it's a really serious problem, the cell knows that there's no way to correct it. So the cell will undergo programmed cell death, apoptosis. Now, if the cell had gone the first route where it would stop and attempt to correct the problem, it'll try to correct the problem, and after some time it will proceed with the cell cycle, whether it has corrected the problem or not. Usually it corrects it and then it'll proceed, but sometimes it will proceed through the cell cycle even though it hasn't completely corrected the problem, and then again it'll undergo apoptosis. And here are some common checkpoints in mitosis. In mitosis, or rather right before the cell enters mitosis, between G2 and M phase. So in G2, the cell has a checkpoint to check for DNA damage and unreplicated DNA. So have I completely replicated my DNA? Are there any errors? Is there any kind of damage in DNA? In the proto-metaphase transition, cells will check whether centrosomes have separated, whether they have moved to the opposite ends of the cell. In the metaphase to anaphase transition, a cell will check whether all the spindle fibers are correctly positioned and where they have attached to the kinetochores. So we're not going to attempt to separate sister chromatids unless everything is correct with the spindle. The spindle is positioned correctly, the spindle fibers are attached correctly to the kinetochores. So these are just some examples of checkpoints. Okay. And in summary, remember, the cell cycle really is a sequence of the phases of the life of a cell. And a cell has 4 phases. 3 of them are collectively called interphase. Those are G1, S, and G2. And mitosis. Mitosis is the actual division. That is when nuclear division happens and cytoplasmic division happens. In interphase, a cell prepares to divide. It grows. It duplicates its organelles. It duplicates its DNA. It prepares itself for cell division. An interphase is also where a cell will actually make the decision whether to undergo mitosis or not. Finally, I would like to say a few words about asexual reproduction, because mitosis is the method of asexual reproduction. Asexual reproduction is the reproduction when you have only one parent and the offspring is identical to the parent, genetically identical. And asexual division can be seen in, can be binary fission, which is a form of asexual reproduction. An organism splits exactly in half to become 2 new identical organisms. And this is seen in bacteria and certain type of fungi. There is also budding, where the new organism is actually like a little bud. It is smaller than the parent organism. And there's cloning, which is a production of identical genetic copies from the parent. So plants reproduce this way using mitosis. So here's the budding. The budding is a type of asexual reproduction. Yeast, sponges, hydra can reproduce this way. A new small individual begins to grow out of the site of the parent organism. It's called a bud. The bud will break free once it's large enough to survive on its own. There's also something called regeneration. Regeneration really is a type of asexual reproduction, and it is a weird, really, mechanism of asexual reproduction. So its main purpose is not really asexual reproduction. Main purpose is regeneration of missing body parts, but it is a form of asexual reproduction. And it can be seen in starfish. It can be seen in certain type of lizards and a type of worm called planaria. And it's really, really weird and really, really cool. Okay, wait. So the planaria is a type of flatworm that it doesn't matter how many pieces you cut it, each piece can grow into a new worm. And that's a form of regeneration. For starfish, really, starfish can lose an arm and regenerate a whole starfish. And that's an adaptation because normally starfish are getting preyed upon by certain type of crabs and lobsters. And as they start chewing in one of the tentacles of the starfish will, they can bite off that tentacle and the starfish will literally flee the predator, missing a limb, missing not really a limb, but a tentacle, an arm, and it can regenerate itself. All right. And I think this is the end of it. So this was it, fellow science nerds. Looking forward to seeing you next time.